So it gives me great pleasure to welcome our next speaker, Dr. Gail Cross, who's joining us from Melbourne. So Gail is a ID physician and TB researcher based at the Burn Institute with appointments at Monash and Doherty Health. And she's worked here in Brisbane as an infectious disease registrar, and I also knew Gail in Darwin in a previous role there as well. And after her time here in Australia, she spent 7 years in Singapore with undertaking a research fellowship with Professor Nick Patton at the National University of Singapore. And she and her family have now moved back to Melbourne, and Gail and I have a competition to see who can finish their PhD first. It's going to come pretty close. We've both got a few weeks till our deadline. So we appreciate you, Gail, taking time to speak to us today about host-directed therapies for TB and do statins have a role. Thanks, Gail.
Thanks so much, Andrew, for inviting me and to the organisers. And I'm sorry I can't be there in person for dinner, but it sounded very good. So I'm going to talk about some work that I did when I was in Singapore at the National University Hospital, and very grateful to the Kirby Institute for taking me on as a PhD student. And I'm going to talk about host-directed therapies for tuberculosis, specifically whether statins have a role. Very quickly, I'm going to talk about what is host-directed therapies in tuberculosis, why we would use a drug like statin, and then talk about the Rosetta trial. So this was a phase 2b trial where we used adjunctive rosuvastatin for the treatment of drug-susceptible tuberculosis. It was a trial where Nick Patton was the chief investigator, and I was his sidekick who ran the trial. And I'll tell you more about it in time to come. Within this trial, we embedded some biomarker discovery sub-studies, of which I'm presenting the data of two today. So PET-CD sub-study, as well as circulating cytokine and chemokine sub-studies. So what is host-directed therapy? Thankfully, I probably don't need to describe this as much as I would have pre-COVID, but essentially it's a drug or an intervention that we use to interfere with host cell factors that are required for pathogen replication or persistence, or the reversal of the prevention of immunopathology due to the pathogen. So the tocilizumab or the baricitinib or the dexamethasone that we use in severe COVID is, of course, a form of host-directed therapy, and we're using it to reduce the inflammation and necrosis that occurs as a consequence of severe COVID-19 disease. And in fact, host-directed therapy has been something that has been thought about for tuberculosis for many, many, many decades. And there are many candidate agents, as we can see on this slide here, some of which really are thought to enhance the host immune mechanisms for killing. And some of that includes statins. And then there are others that limit the inflammation that occurs with TB, particularly in the lung as such a critical organ, as you will all know as respiratory physicians. So why statins as a host-directed therapy? I had to put this slide in because Professor Akira Endo died a few months ago. He was a Japanese scientist who actually discovered the very first statin. He hypothesized that in the 70s, that there would have been some microbes that produced secondary metabolites that inhibited cholesterol synthesis, and they did so as a defense mechanism against other microbes. And his hypothesis was proven to be correct. From Penicillium citrinum, which is your typical mold, blue-green mold that grows on oranges, he isolated the very first statin, which was mevastatin. He used it in people that were homozygous for familial hypercholesterolemia and showed that it was a very, very potent HMG-CoA reductase inhibitor. And as a consequence of that study in the 70s, we now have a plethora of statin drugs. And we don't just use statin drugs to reduce cholesterol and stabilize atherosclerotic plaques. We also are considering it more and more as an agent that we use to reduce inflammation because of its pleiotropic effects, because it also, as part of the same pathway through HMG-CoA reductase inhibition, it also reduces the production of isoprenoids, which are the cell signaling molecules that help with inflammatory signaling. So I'm going to give you a very abbreviated timeline of the association between cholesterol, statins, and TB. In fact, it goes back way beyond 1998, but essentially this was when the first whole genomes was sequenced of Mycobacterium tuberculosis. And in this Nature paper, in the abstract, it's startling that they write MTB differs radically from other bacteria in that a very large proportion of its coding capacity is devoted to the production of enzymes involved in lipogenesis and lipolysis. And I put this here because I wanted to highlight the fact that mycobacteria, and particularly TB, is dependent highly on lipids, including cholesterol. And in fact, in 2000, a group showed that cholesterol is actually required for MTB to be infected, to infect macrophages or to be phagocytosed by macrophages. And that if you deplete cholesterol from macrophages, it actually re-enabled the phagolysosomal fusion and microbacterial killing. And if anyone's interested, I can talk to you about the mechanisms of how MTB actually inhibits that process in a cholesterol-dependent manner. And in 2008, they actually identified the gene that MTB required to import cholesterol into its cells. So MTB isn't an organism that can synthesize its own cholesterol. It's really dependent on importation of host cholesterol into its cell, and then it degrades that to be used for as a carbon source, as an energy source, and as a virulence factor. So from 2008, then, the obvious question was, if cholesterol is this essential for MTB survival, growth, and virulence in animal hosts, and MTB relies on host-derived cholesterol, what happens when we inhibit cholesterol production in the host? And so from 2014 onwards, there were a load of, not a load, but several mouse models that were developed, and it clearly showed that when you use statins, it reduces mycobacterial load in a variety of animal models, particularly in mice, and you could do it on its own or as an adjunctive therapy to standard TB therapy alone. But most importantly, Notan Datta's group, he's at NIH in the state, he actually showed that when you combine statins with standard TB therapy, you could actually reduce the incidence of relapse when you shorten the duration of TB therapy. And of course, at least some of us consider that the holy grail of TB therapy towards elimination may be if we're able to shorten the total duration that is needed for TB treatment. From a programmatic sense, that might actually help with elimination of TB altogether. So very quickly, mechanisms of actions of MTB. Statins can limit the uptake of macrophages. This is what's been shown in vivo and in vitro studies. Statins limit the formation of foamy macrophages, which are crucial for MTB persistence. They limit cholesterol as an energy and carbon source, and they induce phagolysosomal maturation, fusion, and autophagy. And this is the mechanism, the last one, number 4, is the mechanism in which or how macrophages actually kill MTB and therefore exert their host-directed microbial activity. Okay, let's talk about the trial. So this was a phase 2b trial of adjunctive rosuvastatin in drug-susceptible pulmonary TB. It was funded by the National Medical Research Council in Singapore, and the sponsor was the National University Hospital, which is where I worked. The chief investigator was Nick Payton, and I was the project lead for this, and none of the authors have any conflicts of interest to present. So what we did in this trial is we randomized participants to either getting standard TB therapy, which was 4 drugs, rifampicin, isoniazid, pyrazinamide, or ethambutol, that's the control arm, or they received standard TB therapy with the addition of 10 milligrams of rosuvastatin, and that statin was only given to them over the intensive phase of treatment, that is up to week 8, at which point they completed the intervention, and then they continued on the consolidation phase of TB treatment, which was rifampicin and isoniazid alone. At the week 8 time point is when we collected our primary efficacy outcome, which was sputum time to culture conversion in liquid media. And we had a bunch of secondary outcomes, but we also looked at culture conversion in solid media at the 8-week time point. And from a safety perspective, we measured or we collected data on all adverse events, but our primary outcome from a safety perspective was incident grade 3 or 4 EEs, collected at up to the week 8, 24 time point. So our trial was run in Uganda, in Vietnam, and in Philippines. And it ran between September 2020 and January 2021. So we recruited very quickly during COVID, something I'm quite proud of, but it was hard going at the time. We had 304 patients that were pre-screened, and then ultimately ended up with 137 participants that were enrolled in the trial. And of those 137 enrolled, there were 70 in the rosuvastatin group, 67 in the control group. And then, of course, as you always have with trials, one participant had to be withdrawn because, in fact, we discovered that they had rifampicin resistance, which was an exclusion. And in one participant, we could not confirm TB microbiologically. So they got in on a smear, but it turned out that they didn't have TB after all. And then we did the usual modified intention to treat analysis, as well as a per protocol analysis. So between the two groups, 68 and 67 in each, the participants were relatively well balanced. We had a very small proportion of participants who were people living with HIV. About 15% had diabetes. And based on either radiological measures or microbiological, the burden of disease in these both arms were well balanced as well. And here is the main outcomes or the main findings from this trial. So it was published last year in 2023. But essentially what we found was that there was no difference with the use of adjunctive rosuvastatin in terms of time to culture conversion in sputum by the 8-week time point in midget media. And similarly, I haven't shown the data, but in solid cultures, it was much the same as well. But in a subgroup analysis, what we discovered was that there was a signal. Of course, we could not, we weren't powered to show this, but there was a signal that there might have been an effect in the Asian group of patients, that is those from Vietnam and the Philippines, in comparison to our participants from Africa. And we hypothesize that this may be because we know that there's a twofold elevation in the median exposure for rosuvastatin in Asian patients compared to Caucasian populations. We don't know about the African group, but we assume that they might have a different metabolism of rosuvastatin compared to Asian patients. And in fact, this is why there's a ceiling dose of 20 milligrams of rosuvastatin amongst Asian ethnic cohorts, whereas you can go up to 40 in others as well. So within this trial, we embedded a number of biomarker discovery sub-studies, and one of them was PET-CT. So this was carried out with the assistance of Jim O'Doherty, who is a nuclear physicist. He was based at the Clinical Imaging Research Center at NUS in Singapore, and has since moved on to the United States, where he's employed by Siemens. And Siemens had no role in this study, but I'm going to present the data from that. So we did PET-CT scans at 2 time points, very soon after the participants were randomized into the Rosetta trial, and then once again at the 8-week time point. So, because of cost, PET-CTs are expensive no matter where in the world you do them. We had to limit ourselves to being able to do 25 PET-CTs. And because of the accessibility of PET-CT in Uganda, we could only do it in Vietnam and in the Philippines. So we did 25 in total. Unfortunately, one of those that we scanned was the participant where TB wasn't confirmed microbiologically. So we had to remove that participant's data. And that gives us balanced amounts in both arms. And I analyzed 48 scans in total. So Jim helped me with development of a protocol, which, we're hopefully going to publish shortly. Not just the protocol, but the data, but the protocol will be available along with it for anyone who is interested in PET-CT as a biomarker for infection in the lung. Okay, and here are the results from the PET-CT sub-study. So we found that, as we did with the sputum culture conversion, that rosuvastatin did not have an impact on PET-CT metrics. So if we compared the change between baseline and week 8 for the control group and the rosuvastatin group, there was no statistical significance between that if we used total lesion glycolysis. So very quickly, what is total lesion glycolysis? Is on a PET-CT scan, when you look at both lung fields, total lesion glycolysis is the volume of lung that's affected based on CT measures, combined with the uptake of FDG, 18F FDG, so the tracer that you use within that volume of affected lung gives you your TLG. And similarly, there was also no difference between the two groups for SUV max, which is something I think people are more familiar with. that's essentially looking on a PET CT scan is what is the hottest pixel in both lungs, and that gives you your SUV max. So it's a much more blunt tool. And having said that, we then found, of course, that the volumetric indices of TLG performed much better than SUV max as a measure, as a metric that was able to measure the impact of TB treatment. So whilst your SUV max really didn't change between week 4 and week 8, or changed very little and certainly did not change significantly, there was a 64% reduction in total lesion glycolysis even after just 8 weeks of treatment. And this finding is actually consistent with other PET CT studies, primarily done by Cliff Barry's group at the NIH, that have similarly found about a 50% reduction in TLG, although they haven't quantified it so precisely. Okay, so moving on to the other sub-study, which is the circulating cytokine and chemokine sub-study. What we did here was, over the first 8 weeks, we also, at pre-randomization and then at week 4 of treatment, and then week 8 of treatment, we collected blood, we spun it down, and we stored the plasma. And we did this in all 137 participants, but for reasons of costs and logistics, we really analyzed it in 80 participants alone, and that's the distribution across the different sites, Uganda, Vietnam, and Philippines. And so we had 40 in the rosuvastatin group, 40 in the control. We analyzed it centrally, so all these samples had to be shipped from various sites across to the Institute of Molecular and Cell Biology, which is John Connolly's lab, in Singapore. And we decided we'd do, when we wrote the protocol, we decided we'd do a 41-plex assay, which looked at a variety of pro-inflammatory cytokines and chemocytokines associated with inflammation. And here are the results from that cytokine sub-study. So not surprisingly, TB is an inflammatory process, as we all appreciate as scientists and clinicians. And with TB treatment, you get a decrease in the inflammatory cytokine and chemokine profile. And you can detect this within 8 weeks of TB treatment. So there was a reduction in IL-1RA, receptor antagonist, interferon gamma, IL-6, IP-10, GM-CSF, IL-8, TNF-alpha, and IL-10. Those were the 8 cytokines that in a mixed linear effects model that I built decreased significantly over that 8-week time period after adjustment for various other variables. But what was very surprising was that, in fact, in the rosuvastatin group, there was an increase in pro-inflammatory cytokine productions. So when I first saw this data, I reran the analysis and then reran it again, and then presented to my PhD supervisors, who asked me to rerun it again, which I did. And the reason for this disbelief was because really, largely, and I'm sure many here would agree, that statins are largely considered to be agents that have very well-proven anti-inflammatory activity. So part of the reason the pleiotropic effects of statins are thought to be as a means of being able to reduce pro-inflammatory cytokine production. And that was the basis for the very successful Jupiter trial, which was 20,000 patients across the world, where they showed the use of 20 milligrams of rosuvastatin in people with normal cholesterol levels, but with raised highly sensitive CRP. There was a reduction in CRP as a consequence of the use of rosuvastatin that actually was thought to be responsible for the reduction in cardiovascular mortality over a lengthy period of follow-up. And that trial was published in 2008 in the New England Journal. So, and it's consistently been shown that statins are supposed to be anti-inflammatory drugs, and their use largely more and more outside of the cardiovascular space has been precisely for this reason. So why did we find otherwise? But, as you do, you dig a bit deeper, and you in fact find there is quite a bit of data to support your findings. So I have asked this question: are statins pro-inflammatory in the setting of infection? So in early studies in 2000, not just in when you have MTB infection of peripheral blood mononuclear cells or in rat models combined with the statin, there was actually an increase in IL-1B, IL-18, and interferon gamma. So the infection alone, of course, causes a pro-inflammatory cytokine milieu in the host, whether it's the PBMC or it's the mouse host. But with the addition of statins in those experiments, there was a further increase in those circulating cytokines. And then again, I found 20 separate studies that showed that in the setting of lipopolysaccharide or E. coli, whether it was in vitro or in vivo, in the presence of statins, these infectious antigens or these infectious pathogens resulted in an increase in the production of IL-1B, IL-18, IL-6, interferon gamma, TNF-alpha, all the pro-inflammatory cytokines once more. But I guess most telling was the recently, 5 years ago now, published sales trial where you had 745 participants with acute respiratory distress syndrome from lung infection. And what they found was that the use of statins, which is supposed to do the opposite, it in fact increased IL-18 levels, and those with increasing IL-18 levels on a statin had the highest risk of mortality. And I'm very grateful for the fact that, in fact, someone has shown very clearly that when you get microbes that infect host cells that result in inflammasome activation, sorry, I realized my mouse wasn't pointing, inflammasome activation. That inflammasome activation results in caspase-1 activation, and what caspase-1 does is that it proteolytically cleaves pro-IL-1B and pro-IL-18 into IL-1B and IL-18, the active forms of these cytokines, and those are the starting cytokines that lead to progressive inflammation. And what they have shown is that in the presence of something like a statin, you can, in fact, increase or enhance caspase-1 activation in the setting of inflammasome activation. And if you use something like mevalonate, which is downstream of HMG-CoA reductase, so you do a mevalonate rescue, you can, in fact, abrogate that process completely. So, nice mechanistic process of how statins can actually be pro-inflammatory in some setting. Okay, I'll finish up there. Rosuvastatin, therefore, we found had no effect on accelerating sputum culture conversion within 8 weeks of treatment. Is there an treatment effect in Asian versus African cohorts? This is a hypothesis that is deserving of testing. Rosuvastatin appears to have no effect on PET-CT changes over 8 weeks, but TLG is a better metric than SUV max, and it's something that we should be using in PET-CT studies going forward. But I will warn you, it is very difficult to quantify. It takes a lot of time. And finally, we found that statins may increase the secretion of inflammatory cytokines. And we think that this activity may be due to enhancement of caspase-1 activity. Once again, this is a hypothesis that needs to be tested. So lots of people to thank, particularly the participants of the Rosetta trial. I couldn't have done this without Tony Kelleher at the Kirby Institute, Christina Chang, and of course, Nick Payton, who I'm very grateful for taking me on board in Singapore. I received funding from the David Cooper Scholarship Fund at the Kirby Institute, but also through NUS as a research fellow and through the Clinician Scientist Unit at NUHS. And then, as you can see, as with any trial, a huge number of people, very difficult to name all of them. But we had 5 trial sites in total who worked exceptionally hard. Thank you.
Thanks, Gail, and congratulations on your work and that of your team. That's very fascinating. Maybe Professor Shin should put in statin metabolism in his data set.
I'd love that.
Yeah. So you guys can talk to each other. Do you think you are a little bit underpowered to show a difference, or do you think it was a dosing issue as shown by suggested by your different ethnic groups with pharmacogenomics?
Yeah. So I was very keen. So we used 10 milligrams of rosuvastatin, and I was super keen to use 20 milligrams. But it's very difficult to go into a trial, particularly go into a trial where you're only going to use the drug for 8 weeks and start off on a big dose, particularly in an Asian cohort, with the risks knowing that that was the ceiling dose that you could use. So, and in fact, it played out that we should use 10 milligrams because, in fact, we couldn't get ethics approval through in the Philippines unless we started at 5 and then titrated up very quickly to 10 after 2 weeks. So I would have liked to use a bigger dose. I do think that there might have been a dose effect. And of course, we were underpowered. So we were powered for the full trial, but for the sub-studies, because they're sub-studies, they're by nature underpowered. Yeah.
Well, thanks, Gail, for taking your time tonight to speak to us. And
Pleasure.
I'll just mute you now, and I'll go on to our next final speaker.